Today we begin an adventure into the world of bacterial genetics. This is our Unit 4, and in this unit my overarching goal for you is for you to develop an idea of how genetic variation occurs within bacteria, and then how bacteria are able to, as a result of that genetic variation, develop antibiotic resistance. A very important topic when we talk about human health, because we are in the midst of what can fairly soon become a crisis in terms of resistance to antibiotics. You've seen this picture before. This is I think it's an Escherichia coli that has lysed. And all of this curly little stringy material on the outside of that is the DNA that was once all packaged nice and neatly up inside this bacterium. That DNA is a loop. It's very different from our DNA in that regard. It's still double-stranded. It's still a double helix, but it is not packaged together in linear chromatids like ours is. The chromosome in a bacterium is circular. It is looped. And you can see that there are no free ends here. You don't really see any free ends. And that's just one indication, even though you can't see all the outside edges, one indication that that is one nice long loop of genetic material that was once packaged inside that bacterium. We've talked a lot about bacteria. We've talked about the fact that they have been able to adapt to all sorts of environments. There's really not a place on our planet that you don't find bacteria. No matter how extreme the conditions, whether it be intense heat, intense pressure, really high salt concentration, there's very few places, if any, on the face of our earth that you don't find bacteria. So these bacteria are very good at adapting to changing environments. They carry that out. They're able to do that by three major mechanisms. We're going to talk about each one of those during this unit. Natural selection, mutations, and then gene transfer. Some of that is all interrelated with one another, but those are the three major topics we're going to be covering. Natural selection you've heard a lot about in the past. You know a little bit about it based on what you've done in biology. When you looked at evolution, you know that Darwin was the person who sort of threw this out there. You know that organisms that are best suited to an environment are those organisms that are able to survive in that environment, because we've even talked about competition within bacterial colonies, within bacterial populations, and so organisms are always within competition for one another, and those who are the fittest are the ones that survive when times get tough. And that's true not only in bacterium, but in all organisms. The thing that makes bacterium a good model for studying natural selection is the fact that it reproduces so quickly, we are able to actually watch it happen over the course of, a very short time period. You can get hundreds and thousands of replications, where it's kind of hard to do that with humans or elephants or trees. So bacteria are good organisms to study for that. The thing about natural selection is those organisms that are the fittest, that are the most well suited for their environment, are the ones that live long enough to replicate and then pass on that gene or set of genes that makes them able to survive in those environments. So important thing about survival is the fittest is the fact that those organisms best suited to survive are the ones that live long enough to replicate and then transfer those traits, those positive traits, onto their offspring. Let's do a quick review of a couple of key terms you need to know about when we start talking about bacterial genetics. You learned about these in biology, so this should definitely be a review. When you talk about the genotype of an organism, you're talking about the actual genetic code, the adenines, thymines, cytosines, and guanines, the allele that codes for a specific trait. Think back to biology when you did genetics and you had a dominant allele and a recessive allele. Those things are the genotypes. Those are actually written into the DNA. That is what is written in the DNA. The phenotype is the expression of that genetic code. I always tell my kids, physical characteristics starts with a pH, phenotype starts with a pH, and that's how you can remember which is which. The physical characteristic of a bacterium would be evidenced by the genus and species to which they belong. Genetic change in a bacterium can be dramatic. The change in one simple gene can dramatically change a bacterium because, we've talked about this before, they are haploid. They only have one copy of the chromosome in them. Very different from us. We're diploid, remember? So you have the ability for instance, a dominant gene to cover over a recessive gene. That doesn't happen in bacteria because they're haploid. So if there is a change in the genetic material in bacteria, then it can very easily alter those observable characteristics, that phenotype of that bacterium, because there is no other copy that might cover up, whether it's a mutation as a result of natural, just a mistake in the DNA replication, or if it's something that has been induced by environmental factors. It doesn't matter. There's only one copy, so there's nothing to cover it over. So therefore, a change in the genotype in a bacterium is very likely to change the phenotype. Doesn't always happen, but it certainly is much easier to do that in bacteria than it is in a diploid organism of any kind. When we talk about genetic change in bacteria, there's two overarching categories of transfer of genetic material among organisms. The first is a vertical gene transfer that you're sort of familiar with. We'll talk about that in a minute. The second is a horizontal gene transfer. Those are your two big types. We're going to look at ways each of these can happen, but you definitely need to know these two terms, and you need to know what falls into each of these categories. Vertical gene transfer is when a mutation arises in a single cell and is then passed down to its progeny. Actually, you don't even have to be talking about a mutation when you talk about vertical gene transfer. It can also be just the standard transfer of genetic material from parent cell to offspring or progeny cells. But because we're talking about change, that's why I threw in that term mutation. But remember binary fission. You've already studied binary fission. You know that binary fission is a form of asexual reproduction, and what you get as a result of binary fission is two cells that are genetically identical. There's something that has to happen right before binary fission in order for that cell to be able to divide into two cells that each have an exact copy. And you should know what this is, but just in case you don't, I'm going to tell you. It's the DNA replication. DNA replication has to happen right before binary fission occurs. The question you might have is this. If you've got these bacterium that are genetically identical to one another, then how are you going to get bacteria that can adapt to their environments? If they're all the same, wouldn't they all react the same to their environments? Yes, they would. So the thing is, Vertical gene transfer, binary fission is not the only thing that happens in bacteria. There is a way that bacteria cells can develop genetic variation, and you're going to learn about that in this chapter. Horizontal, sometimes some books call it lateral, so you may see that when you get to college. We're going to talk about it as horizontal gene transfer. But this is when a cell can acquire DNA, a piece of DNA, a gene, a strand of genes, a section of a chromosome, whatever, from any other source, whether it be another cell, from it free floating out in the environment, or when you talk about lab research, you know, scientists are able to actually implant pieces of DNA into bacterial cells and then be incorporated into the genetic code of that bacteria. So any of those things, getting DNA from any other source, is called horizontal gene transfer. Once that happens, then that change or that mutation is passed down through all of its progeny, just the way it always has been. And you'll see this in a diagram. Here's a diagram of vertical gene transfer. Look at just the top half of this diagram first. Okay, we have this. Now, what we have in this cell right here, you have a group of cells. You have 3 cells in this diagram, and the red is the DNA of that cell. Now, in this picture, it shows this little green piece right here represents a spontaneous mutation within that DNA. We'll talk about how all that can come about later. Let's just take it at face value for right now. That little green piece is a mutation. These don't have it. The one on the right and the left don't have it, just the one in the middle. When this cell takes that DNA and replicates it, that mutation replicates also. And then you will find it passed down into its progeny. You see the little green mutation here. You see the little green mutation there. And then when those cells divide, again through binary fission, you also see the little green mutation in the progeny of that cell. So that is vertical gene transfer. Okay, it happens all the time during binary fission. But when you're talking about a mutation that just spontaneously occurs within the cell, then we term that vertical gene transfer. The second part of this diagram shows genetic material coming in from an outside source. This is one of the ways that can happen. A plasmid can actually be transferred into a different bacterial cell. So you have one cell here. Remember back when I had you draw the bacteria cells early in the semester, I had you include the plasmid, even though you didn't know what it was for yet. I told you it would be very important to us later. Well, we're there. This plasmid has some important genes on it we'll talk about in a little bit. But horizontal gene transfer is where this plasmid makes its way into a different cell. It doesn't have this plasmid. See, there's no plasmid over here. But once horizontal gene transfer happens, then this cell does have the plasmid. So that's horizontal gene transfer, just like the horizon going side to side. Once that genetic material has been incorporated into the cell, then vertical gene transfer occurs just like it did up here. But this is horizontal gene transfer, where you've got genetic material coming in from an outside source. And then once it's incorporated, it is passed down to its progeny through vertical gene transfer. Genetic mutation occurs within the DNA of an organism and then changes it. So the wild type organism is the strain of any particular bacterium that is very similar to what we originally found, what we originally discovered. That's wild type organism. if you get a change in that, anything in the base sequence happens so that it takes on a different phenotype, then it is no longer considered to be a wild type organism. Um, that's just a key term that you need to know. And that's where we're going to stop today.